Hello, and welcome back to Biology, Human Evolution. Today we want to talk about genes and inheritance and all that fun stuff. Because really, when you're talking about evolution, you're talking about passing on genes from one generation to the next. But first, I just wanted to show you this. I think this is wonderful. This is a karyotype, and this is a human karyotype. And you can see that you have all 23 pairs of chromosomes laid out, so you can see what they look like together. And this actually is from some individual. That individual is female because you can tell you have the 23rd pair, which here you have two X's. If it were an X and a Y, it would be a big one and a little one. But anyway, I just think that that is a really cool thing that you can actually take the chromosomes and match them up and line them up and look for problems, right? Which brings us to your first question. What is not normal about this human karyotype? There are too many chromosome pairs. That could be a problem. There are too few chromosome pairs. Also a problem. There are 3 number 21 chromosomes, a problem. The sex chromosomes are not paired correctly, also a problem. So this one, well, the sex chromosomes look fine. You've got the X and the little Y there, so this is a male. You have 23 pairs, you can see, count them all out. So there aren't too many, there aren't too few, but if you look here at number 21, you have an extra little chromosome here. This causes real problems. This is trisomy 21, which is also known as Down syndrome. And we're going to come back to talking about this, but this goes to that idea of talking about chromosomes and chromosomal mutations and things like that. And I just wanted to remind you, since we're going to be talking about individual genes, that the chromosomes are important too. Okay, but now to move on to our chromosomes and genes conversation, this is actually the human genome, and this is a map of all the genes that we knew in 1990, and of course we know a whole lot more than we used to. And you can see that the chromosomes have banding patterns, and this is the X chromosome, and these are actually regions that have genes for all of these different genetic disorders on the human chromosome, and take a look at that. I think it's really interesting, but these all come with just the X chromosome. So imagine when you start looking at all the other chromosomes, all the genes and possible disorders that we might be talking about. Okay, now the variety that comes into play, because one of the things that we talk about with evolution is, of course, these pressures acting on variation. The variety is caused, we know, a little bit by mutation, but we know mutations are few and far between. So really the most important mechanism for variety is genetic recombination, and that's going to be for sexual reproducers. The idea that you get one set of chromosomes from the male and one set of chromosomes from the female, and then there's another little thing that happens that we're going to talk about in a second, but you get this great variety just by having two parents. Okay, so you've got your sperm, your egg, it fuses, and then you get a nice little variant from the original parents. Now, in mitosis, which we've talked a little bit about when we're talking about places that you can get mutation, we know that in the synthesis phase, if the DNA is not copied properly, that we get mutations, and that can be a problem. But then here we have the actual cell division, and we know we can get weird chromosomal anomalies like Down syndrome when we have the chromosomes failing to split properly, and that's all very exciting. And then here's just the cycle, the cell cycle that we go through during the mitosis phase. And we start out with prophase, where the DNA condenses, the chromosomes become visible. That's what we put into the karyotype. In metaphase, they line up. In anaphase, they split apart to either end of the pole. In telophase, they start to decondense the nuclear membranes. New nuclear membranes start forming, and cytokinesis begins. Cytokinesis is not part of mitosis. It's its own thing, and it is the division of the actual cell membrane. And this is mitosis in a nutshell. Now, meiosis, though, this is the part that really is going to contribute to the genetic variety, but I wanted you to understand mitosis first. Now, DNA is copied in meiosis as well. You can have replication errors there and get mutations. You can also get chromosomal anomalies by having them not split properly. That also happens in meiosis. But the thing about meiosis is that the end product of meiosis are going to be the sex cells, right? You either get one egg and 3 polar bodies, or you get 4 sperm, depending on whether you're male or female. And in this, you have the same thing. You duplicate the DNA just like we do in mitosis, but a fascinating thing happens in prophase 1, in that first phase of meiosis. And meiosis actually goes through two kind of rounds, right? So the first one, you have the duplication. That's great. And then you go through all of the prophase, metaphase, anaphase, telophase, and then you do it again, right, in order to get the next guys, except you don't duplicate the chromosomes again, okay? We're going to talk about that in a second. But the one thing I want to talk about first is the crossing over, all right? When the chromosomes pair up, right, so they get the homologous, the sister chromatids, they pair up, they will actually cross arms and exchange DNA. And we talked about this when we talked about mutations and chromosomal mutations. This is a point at where you can get some pretty serious chromosomal mutations. But if you don't, if it works normally, then you just have this little exchange of data. So, or genes, right? Data, genes. So you have now, instead of having a chromosome that was just like this one, these two are mixes of this chromosome, and that is going to give you some traits that might arise that you didn't have in the parents, simply because of this shuffling of genes, if you will. Okay, and here is actually a good picture, like a real picture of the DNA crossing over. You can see all the coils, you can see the chiasma forming. These are these are the centromeres. So you can see the chiasmas forming. This is where they're actually crossing over. They're the centromeres. I have to actually watch where my mouse is. And then these are going to exchange, right? So you can see here when it's done in the false color, the red arm is going to switch with the blue arm here, and then you'll end up getting something that looks like this, yeah, which is pretty exciting. And here's a really exciting one. You can see that the centromere is here in the center, and then you have these chiasma, lots of them, right? This is called a tetrad, and this is actually crazy crossing over, right? And then here are the pairs. These are the things that are going to be crossing over. This, I just put in the male-female thing again, okay? But just to get back really quickly, we're going to back up just for a second. So there's where the crossing over. Then you split, okay? But eggs have to have half of the chromosome set, and sperm have to have half of the chromosome set. So you're going to do all of that again and split again. And so you get a haploid cell, right, as opposed to a diploid cell. So the end product of mitosis is an exact replica of the original cell. The end product of meiosis is not. It's four little half cells, if you will. They have half of their full cells, but they have half of the chromosomal complement that a diploid cell has, right? So eggs and sperm fuse to make a diploid cell. Okay, so let's get back through all of this again. To your next question. An allele, right? And this is we are talking about alleles, allele frequencies, alleles in relation to phenotypes and genotypes. That's what we're going to be talking about now. Okay, so we took those karyotypes. We know that they have genes on them. That's very exciting. But they also have alleles. So what is an allele? Is it another word for a gene, a homozygous genotype, a heterozygous genotype, or one of several possible forms of a gene? Well, an allele is a version of a gene, so D. Okay, so now you have your chromosomes. Well, each chromosome, while they have the same genes, they may have different versions of the gene. Here you have allele A, okay, and here you have allele B. So these may actually code for different things. So like flower color is a really good example, okay? And anywhere where you are looking, where you have a gene, that is called a gene locus. If there's more than one, they're loci. Here, right, we've got a dominant allele and we have a recessive allele. So when we talk about dominant and recessive, we're talking about one that's expressed, and we're going to talk about that more in just a second. But when you start looking at the chromosome pairs, you're talking about the genotype, right? Each genotype, yeah, and that is the gene combination that gets expressed, is going to consist of two alleles, right? Those alleles may be the same, they may be different, okay? And so here you can see that if they're the same, You've got two dominants, you've got two recessives, and you have one dominant and one recessive. And again, we're going to talk about that in just a second. But first, an individual who has two of the same alleles is said to be homozygous, heterozygous, homologous, heterologous, which is not a word, or diplozygous, right? They are homozygous, right? If you have two of the same allele, you're homozygous. If you have two different alleles, you're heterozygous. And those are the two terms we need to know. So let's say that you have these two cute little mice. Both of these mice are heterozygous. You have one dominant allele and one recessive allele. One dominant allele, one recessive allele. There is a 50% chance that either of these chromosomes will go into the egg, right? There's two of them. They split apart, 50% chance, okay? So here's an egg. Let's say that this egg got the dominant allele, and this sperm got the dominant allele. When they get together, then the baby mouse will have two dominant alleles. They will be homozygous dominant. Now, over here, you have the dominant egg, right? But this sperm got the recessive allele. So this one is going to be heterozygous, just like the parents. Same thing over here, because you got the dominant from the male and the recessive from the female. This one is going to be heterozygous, just like the parents. Now, this little guy got recessive, and interestingly enough, he is different, or she is different, because you can see what the recessive allele actually does when you have two recessive alleles. The recessive allele will not be expressed unless it is homozygous recessive. Okay, it won't be fully expressed. All right, so here's your next question. The offspring of two parents that are heterozygous for a given trait have a what % chance of being homozygous for that trait? Okay, well, if you go back to this, these guys are heterozygous for black, okay? But if they are going to be homozygous for black, there's only one combination. Homozygous dominant gives you homozygous dominant. 1 in 4, or 25%. There's 25% that it will be homozygous recessive, and there's a 50% chance that they will be heterozygous. Okay? So the answer to this question is 25%, and we're going to do more. Don't panic. Okay, so let's say that we take two true breeding flowers. A true breeder is a homozygous. Okay, and so you've got homozygous dominant here and a homozygous recessive. They will each give one allele. They only have one type to give, so it really doesn't matter which one they do. And then their offspring will get one of each, and this is a heterozygote. Okay, heterozygous flower. So, right, then we can cross these heterozygous individuals. So here we've got heterozygous. They're going to give their gametes, okay? And so you get, P was a really unfortunate choice here, but this is a capital P, lowercase p, capital, lowercase, capital, lowercase, capital, lowercase. So here you have the same thing that we just saw with the mouse, and here are all the words that describe it. These are all the things I just said, but they're for you in writing. And so now, right, we have this 1 to 2 to 1 ratio, right? And so this is actually the Punnett square that goes with this. What you are looking at, by the way, this drawing of this chart is called a Punnett square, okay? And we're going to be using them a lot. So, the appearance resulting from the given gene combination, or genotype, is referred to as the genotype, phenotype, allelotype, haplotype, or stereotype. It is phenotype. The genotype is the gene combination. The appearance is the phenotype, okay? And genes can combine, right, to give the entire picture. In this case, we have a gene for round, right? And we have a gene for yellow. So we have a gene for texture and a gene for color. So R is round, Y is yellow. Now, you'll notice that we've got round peas and wrinkle peas, but we only use the one letter. That is because each gene is characterized by one notation. So if you have a capital R, that is a round. So the dominant, whatever it is going to be the notation. Okay, so round is dominant, but then wrinkled will be little r, not W. Okay, it won't be W, it'll be a lowercase r. Green is not going to be G, it's going to be a lowercase y. And I put this in here, this is called a dihybrid cross. The other ones we were looking at were monohybrid crosses. Just to show you that this can get really complicated really fast. And if you start talking about human genes, we've got 19,000 to 20,000 genes. You could imagine that the Punnett square for that would be ridiculous. Okay, just going to say it. All right. Now, we were talking about traits that have complete dominance, right? If you look at this, right, the one we had before, you only have two phenotypes, right? The homozygous, there it is, the homozygous dominant and the heterozygous are both the same. This is called complete dominance. But not all genes, not all alleles are completely dominant. Some of them are incompletely dominant, and in that case you get three genotypes. So we have the same idea. We have two true breeders, and they produce heterozygotes. But in this case, because it's incomplete dominance, you have red and white making pink, and this happens with lots of things. Complete dominance is two phenotypes, the dominant and the recessive. Incomplete dominance is three phenotypes, the homozygous dominant, the heterozygous, and the homozygous recessive. Okay, so those are two different situations, yet the ratios and the behaviors of the alleles are the same. Okay, the distribution, the inheritance. Okay. What if, though, you had a gene that had two dominant alleles, and those could be expressed at the same time? This is actually something that turns out to be important to you, and we'll talk about why a little bit later. What is this called if you have two dominant alleles? Codominant, incompletely dominant, mutational, or polygenic? Well, polygenic means multi-genes. We're talking about one gene at a time, so that would not be it. Mutational, no, because that's totally different. Incompletely dominant, we just talked about that. That just means that your dominant allele is not fully expressed when it's combined with the recessive allele. Codominant is when you have two dominant alleles. And here is a really good example of a codominant situation. Okay, you have the pink and you have the white, and they are both dominant, and so when they're combined, you get a little splotchiness of both, right? Here's another example, roan horses and then coloration in roosters. You can see that this is blue, this is white, and when they are together, you get blue and white, the codominant, okay? And then roan horses are the same way. This is going to be red and white, makes a strawberry roan, right? Same idea. Now, you can take that same concept with complete and incomplete dominance and multiple dominant genes, and then you can start adding other genes in. So you have these cute little Labradors, okay? Labrador retrievers, labs, adorable. You have black, you have brown, and you have yellow, right? And they are caused by the interaction of two genes. This is a polygenic trait, okay? So you can either be black or brown if you're a Labrador retriever, or you can be yellow. Yellow means that you have a gene that does not allow for pigment. This E right here is color or no color, right? And then if you have the color gene, if you have a dominant, they're completely dominant, so you either have color or you don't have color. If you do have color, then there are two color types, the completely dominant black or the recessive brown. And so this is where you have those interacting together. And of course, this can start to get really complicated. In this case, you have several versions of the allele. Okay, they're all some are dominant, some are codominant, some are recessive, some are more dominant than others, less dominant, whatever. And then you end up getting like, oh, let's add in spots or, you know, whatever. And so you start getting these really interesting combinations that become really complicated really quickly. Here is horses again, right? So you have the same idea as the labs. You have a base color. You either have it or you don't, okay? But then you can start adding in other colors, like maybe you have a darker mane and tail. Maybe you don't, right? Here, you don't have any change, right? Here you have some variation. Then you can dilute the color, and so you get even more variation on that, right? All kinds of crazy stuff there. And you could get them all at the same time. Hey, that's kind of exciting. That is crazy coloration. Now, all of the things that we've been talking about individually are Mendelian traits. They may act together so the overall outcome is polygenic, okay? And that is something we're going to talk about more next time. But all of these are, you either get the allele or you don't get the allele. So you have the completely dominantly expressed trait. It's either incomplete dominance or it's codominance. Now, there are lots of traits that there are other factors going into it, but we're not going to talk about any of those. So I put in here this little list of Mendelian traits that you can see if you have, right? And so the dominant genes are on this side and the recessive genes are on this side. So go and see if you can roll your tongue, if you have freckles, dimples, arches, widow's peak, double jointed thumb, whatever, okay? All right, now the last thing I want to talk about really quickly is lethal alleles. Okay, sometimes combinations do not work out very well, right? And the classic example of this is mice, and specifically yellow mice, right? So when you have these little yellow mice, okay, you have these heterozygotes, okay? When they are homozygous recessive, they're white. When you have a heterozygote, they are yellow. When you have a homozygous dominant, they don't develop. Okay, so what this means is that the gene that codes for the color is somehow affecting the biochemical pathway for development in the womb. Okay, so you have these two different things, but one gene actually has impact on both of those things. This is called pleiotropy. When a single gene affects more than one biochemical pathway, that is pleiotropy. And it turns out that you don't get any color at all because before you can even get color, this gene is affecting development and making it not happen. And so you get no mice. A quarter of them, you can project out a quarter of them will die in the womb. Okay, so pleiotropy. It doesn't always have to be lethal. There are all kinds of examples of this, but it can cause real problems sometimes, especially if you're combining the lethal alleles with the pleiotropy. Okay, that's it for this conversation. If you have any questions, please ask me next time, and I will see you in class. Thanks for listening.